Hi everyone. In the previous part, we had discussed about the conventional methods, which is one of the type of detection of pathogens in the food. In this part or in this video, we are going to discuss about the rapid detection methods in detecting the pathogens in the food. So, what is a rapid methods? And in general, any method that detects a foodborne pathogen in a shorter period of time as compared with the conventional method is going to be considered as rapid method. That is, they are able to detect the presence of pathogens in raw and processed food immediately as early as possible. These rapid methods are also going to be of very sensitive enough to detect the pathogens which may be present in low numbers in the food sample. The advent of gene probe techniques has allowed the development of powerful tests by which particular bacterial strains can be rapidly identified without the need for isolating in the pure culture that we will do in the conventional methods. And the molecular based methods can detect the viruses in shellfish but are not yet available for other foods. To what is the need of this rapid analysis? Why we are supposed to have the rapid analysis or rapid detection methods? That number one, to prevent the outbreaks of foodborne diseases. That means if you are going to detect the presence or the cause of the particular type of the foodborne disease after 3 to 7 days, meanwhile the person may die or it may spread more. So for that reason, the outbreaks can be prevented by using this rapid detection methods. Number two, to provide the immediate information on the possible presence of pathogen in raw and finished products. So once you are going to give the immediate cause of that particular type of the foodborne disease and detecting the pathogen, immediately the doctor or the medical person can prescribe the medicine regarding that pathogen. Then it is going to reduce the human errors that is occurring in the conventional, so that cannot be occurred in this one. Then validation is going to be more, automation and computerization, and simplicity in handling, and to save time and labor cost. So we can save the time also. So that means the presence of even a single pathogenic organism in the food may be a infectious dose. So we can detect even the presence a single or very low number of the pathogens that are present in the food sample by using this rapid detection methods. That's the need of this rapid analysis. Then what are the different types of the rapid detection methods? Means we are having mainly three types. One is the immunological methods or antibody assay methods. Then second one is nucleic acid based methods. The third one is the biosensor methods. So under the immunological methods, we are going to have the latex agglutination test, ELISA, lateral flow immunoassay, reverse passive latex agglutination test. Coming to the nucleic acid based, we are having the PCR, that is a polymerase chain reaction methods, then oligonucleotide DNA and microarray, that is DNA microarrays. Then biosensors, we are having the different types of the biosensors which are used in detecting the pathogen, that is optical based, electrochemical based, immunosensors, piezoelectric biosensors, and surface plasmon resonance biosensors. So out of all these things, we are going to discuss few about each type of the methods now. Let's begin with the first one, that is immunological methods. So the immunological detection methods, which are also called as antibody based methods. So here, these are going to facilitate the design of variety of assays and formats. And the incubation times are going to be very short for methods such as agglutination reactions commonly used for the rapid identification of organisms. That means here the antibody is going to be labeled with a fluorescent reagent or with an enzyme so that the antigen antibody interaction may be visualized more easily when it is going to occur. So fluorescent microscope technique in finding out the antibodies is the best example. Then in some cases, the antigen antibody complex formed is directly measurable or even visible. So like in the case of latex agglutination. So these antibody techniques or highly specific interactions of antigen antibody are going to be used for detecting the pathogens. So here we are going to have, I gave the list: latex agglutination, reverse-based latex agglutination test, immunodiffusion, ELISA, that is enzyme-linked immunosorbent assay, then immunoprecipitation, lateral flow immunoassay. So coming to the first one, latex agglutination test. The use of latex agglutination test is going to be of having the detection of antibodies against a protein that is present in the organism or any substance that the bacteria produce. Example, if we check a rabbit agglutination test for Staphylococcus aureus, then this Staphylococcus aureus is going to have a protein A and the receptor for fibrinogen. So the latex contains an IgG antibody capable of binding to a protein A and a receptor for fibrinogen. Now, if the sample is going to have this bacteria, that is Staphylococcus aureus, now this IgG antibody binds to that protein A and you are going to have this agglutination. If that sample is not going to have the presence of this Staphylococcus aureus, so obviously you are not going to have this one. That means no agglutination will occur. So that's how the agglutination is going to indicate the presence of the bacteria or the contamination in the food. Then moving to the second type, lateral flow devices. So here the lateral flow devices are going to be comprised of a simple dipstick made of a porous membrane that contains colored latex beads or colloidal gold particles coated with detection antibodies targeted towards a specific microorganisms. So this may be a ready-made type of the one. The particles found on the base of the dipstick, which is put in contact with the enrichment medium. Okay, if the target organism is present, then it will bind with the colored particles and it is going to find the what we call as a colored line that is clearly visible in the device window, indicating a positive result. As with other immunoassays, the LFD also requires some sort of a previous enrichment, and the technique is going to be extremely simple to use and easy to interrupt. That means you are going to place the sample here and it slowly diffuses through this nitrocellulose membrane, and during the process it may have the conjugate pad, and now if it is going to detect that conjugate one, it will move, and the concerned antibodies, if they are going to detect, they will going to have the reaction, otherwise you won't see any test line positive. So there are various types of LFD, that is lateral flow devices, that are available in the market, like that sandwich assay format, which is going to be used for analyte proteins with multiple antigenic determinants. And in the same manner, we have another type of LFD, that is competitive assay format, used for small molecules like hormones and the drugs, etc. So here you can see the picture how a sample is going to be diffused in the lateral flow assay. So the sample, this is an analyte that we want, and it is going to have the conjugate moving through the conjugate. If it is positive, it will bind, and through the capillary flow movement, it is going to move towards the test line and the control line, which is going to have the coated with the antibodies. Now this analyte, if it is positive, it will bind to this one along with the conjugate, which makes the thing to be faster, and you will see the red line here, positive line. If not, you won't see any color here because it is not specific. Okay, so this is all about the lateral flow assay or device. Then moving to the ELISA, enzyme linked immunosorbent assay. It is a biochemical technique, obviously, which is having a combination of immunoassay with the enzymatic assay. So an antibody bound to a solid matrix is used to capture the antigen from enrichment cultures, and a secondary antibody that is going to be used here is conjugated with an enzyme which is used for the detection process. That means this enzyme is going to have the conjugation with the secondary antibody, will bind only to the antigen antibody complex. If an antibody is there, if the sample contains the antigen, then obviously both will form a complex. Then if they are not specific to one another, there will be no complex formation, and the enzyme also won't bind, because this secondary antibody or target analyte is going to bind only to the antigen antibody complex. And if this is present, we are going to add a substrate which is going to lyse it and give the colored product. So that color is going to be automated in the ELISA reader, and they are going to detect the concentration of the concerned antigen or antibody, and the result can be obtained within 2 to 3 hours instead of taking the days together, which is going to be done in the conventional method. So we are having the different types of ELISAs, that is indirect ELISA, sandwich ELISA, and the competitive ELISA. So in the indirect method, the antigens are going to be coated in the well, and then the antibody was specific antibody to be measured from the sample is going to be added. If they are specific, you are going to bind to the antigen antibody complex, and secondary antibody will go and bind to this what we call as a antibody antigen complex. And that's how, if once you add the substrate, you are going to have the color giving that means lysing of the substrate will give the color, and that lysing of the substrate is going to bind done by this enzyme. Okay, so the detailed explanation about the ELISA I have given in the immunology subject ELISA. So there you can go through it, where the detailed explanation of this ELISA I have given there. Okay, then moving to the sandwich ELISA. So here it is going to be a combination of two antibodies. That means the antibody first you are coating, then antigen you are adding the sample, and then the secondary antibody is going to bind. So as the antigen is in between, we call it as a sandwich. So in the competitive, there will be a competition of the between the specific antibody and the unknown antibody. So if the specific antibody is going to be there, obviously it will bind to the antigens, and if it is already occupied, it won't bind. So there will be a competition between these two antibodies to attach to the antigens. So if the mixture is going to have the more antibodies, then it is going to bind these antigens and it will cover. So if it is not going to be more number of this one, then obviously it is not going to do. So this is how we are going to detect the pathogens or the concerned antigens in the food samples by using the ELISA method. Then moving to the second type of the methods, that is nucleic acid based or DNA based. So here these includes especially the nucleic acids for the detection of the pathogens. Previously, we tried to find the antigen, specific antigen or antibody, and in the second method, we are going to have the methods based on the DNA. So, what are those things? Means DNA hybridization, then PCR, that is polymerase chain reaction, then DNA microarray. So here coming to the DNA hybridization. So in this DNA hybridization, the DNA probe sequences of known segments are hybridized with the target genome. If the relevant sequence is there in the sample, the probe binds with that portion and gives the fluorescence. And the target DNA is denatured at the high temperature. Now the probes are labeled with the radioactive labels for detection. Now, through the auto radiography is then used to detect the probe target complexes. That means whatever we are going to send some signals, and those signals, if they are going to bind with that concerned one, so obviously you are going to have the detection of those probes, and with the known probes we are going to detect whether the pathogen is present or not. So as it is a hybridization between our probe and the concerned nucleic acid of the pathogen, we call it as a DNA hybridization method. Then moving to the PCR. So here the PCR is going to be a special type of the technique used for the in vitro enzymatic synthesis of specific DNA sequences by tags and other thermoresistant DNA polymerases. And these PCR technique uses oligonucleotide primers that are usually 20 to 30 nucleotides in length and whose sequence is homologous to the ends of the genomic DNA. So PCR technique method is performed in repeated cycles so that what is going to be the products of one cycle serve as a DNA template for the next cycle, doubling the number of the target DNA copies cycle. So here you can see. So the rapid increase in the number of copies of the target sequence can be achieved with the PCR based methods and makes them ideal candidates for the development of faster microbiological detection systems. So many PCR techniques are going to be having a validity and commercialized to make the PCR a standard tool used by food microbiology laboratories to detect the pathogens in the foods. So there are this PCR technique is going to be having the three main processing steps in the one. The first one is the denaturation, the second one is going to be the annealing, and then extension. In the first step of the PCR method, the DNA is heated to 95 to 98 degrees centigrade, at which point DNA gets separated into single strands. Next, in the annealing step, the synthetic oligonucleotide primers are added. That means we are adding the primers are added and bind to the single strand at a temperature of about 55 degrees centigrade. Then finally, in the last step of the process of primer extended by DNA polymerase in the presence of an adenine, guanine, cytosine, and thymine. So here you are going to have the extension of this strand. Now, once the extension is going to be done, the three steps are going to result in the new DNA strands that are going to contain these sequences. So this is a brief note of this PCR technique. Now, there as the fluorescent dyes such as in the real-time PCR method. So that means we are going to have so many methods. So the conventional PCR, real-time PCR like this we are having. So this method is going to be more specific and rapid than our conventional methods like enumerating SPC, then MNC, all these things. Okay, so the conventional PCR relies on amplification, that is amplification of the target genes in a thermocycler and separation of the what we call as PCR products by gel electrophoresis, followed by visualization and analysis of the resulting electrophotographic patterns. That is a process that can take a number of hours. The specificity can be subsequently confirmed by sequencing the amplified fragment. So the real-time PCR is a type of or advanced type of this normal conventional PCR, where it is going to allow both the detection and quantification of the single emitted by the amplified product by using a continuous measurement of fluorescent labeled during the PCR reaction. The increase in the fluorescence can be monitored in a real time, which allows us to have a accurate quantification over several orders of magnitude of the DNA target sequence. So that's how. The real-time PCR is going to be used for viral quantification, drug efficacy, then pathogen detection, then genotyping. Then moving to the DNA microarray. So here the DNA microarray is going to be the core principle behind the microarrays is hybridization between what we call as nucleic acid sequences or DNA strands. The DNA or the target sequences are bound to a chip, which is normally a glass slide or a nylon membranes called arrays. Now the mRNA is going to be extracted from a control and the experiment cells are labeled with the specific dyes. So here we are going to have the specific dyes like a, that is CY3 and CY5. And then they are going to be hybridized with the target probes attached to the arrays, and this is going to be of a rapid and efficient method and can detect thousands of the specific sequences. So here you can see the computer analysis, a combination of these two arrays are going to be seen in the yellow color. Then moving to the next one, the biosensors. So the biosensors are going to be defined as the indicator of biological compounds. That means the biosensing methods for pathogenic detections are centered on four basic physiological or genetic properties of microorganisms. Number one, metabolic pattern of substrate utilization. Then number two, phenotypic expression analysis of signature molecules by antibodies. Number three, nucleic acid analysis. Number four, analysis of the interaction of pathogens with eukaryotic cells. So here these biosensor devices are going to convert a biological response into the electrical signal. So where you are going to see. And this consists of a bioreceptor element and as well as a transducer. And this transducer is going to convert the signal of electrical current and passes it to the amplifier, where you are going to see the light or the whatever the signals. So we are having the different types of the biosensors like bacterial bioluminescence, then fiber optic biosensor, then we are going to have the electrical impedance biosensor, then piezoelectric biosensor, and ATP bioluminescence. So here, the first thing that we are going to discuss is about the bioluminescence biosensor. It is going to measure the change in the luminescence emitted by living microorganisms. So there are certain microorganisms which are having the capacity of luminescence, that is evolving the light from their body. So that is going to be called as bioluminescence. So this bioluminescence is going to be of found in microbes is two types, ATP bioluminescence, and another one is going to be the bacterial bioluminescence. So first one is going to be the ATP bioluminescence. In this technique, it is going to measure the emission of light produced by an enzymatic reaction between two enzymes, that is luciferin and luciferase, that requires the presence of ATP. So that means the ATP test is going to be a process of rapidly measuring actively growing microbes through detection of adenosine triphosphate, that is ATP simply. As ATP is a molecule found in and around the living cells and provides a direct measure of biological concentration and health, because ATP is a primary source of any living organism, even the microbes also. So the ATP disappears within 2 hours after the cell death in any cell. So once the cell is going to be dead, the ATP also disappears within 2 hours. So in this whatever the ATP bioluminescence experiment, here the ATP is going to be quantified by measuring the light produced through its reaction with luciferase enzyme. So here I told you the luciferase, an enzyme naturally occurring in firefly, for example, using an instrument called as a luminometer, and the amount of light produced by directly proportional to the amount of living organism present in the sample. Technically, the existence of ATP luciferase emits what we call as a light, which is measured with a liquid scintillation spectrometer or luminometer immediately. Okay, now the main advantage of this one is going to be of a simplest and rapid ways to measure the ATP level in the food sample, and hence helpful in estimation of microorganisms in the food and the food products. Okay, so mainly that come in contact with the food. So if you see the reaction here, the luciferin is going to have the utilization of ATP and gives rise to a compound called as lufiryl adenylate, and it requires the ATP. That means it is going to break the ATP, and this one reacts with the oxygen. That means as the organisms is going to take the oxygen, the luciferyl adenylate in the presence of oxygen gives rise to an oxyluciferin compound, as well as the light, the emission of the light, which we are going to find it with the luminometer. Then bacterial luminescence. So the genes responsible for any bacteria to give this bioluminescence or light is going to be called as a Lux gene. The Lux gene has been identified and it is going to be cloned. Now, the DNA carrying this gene is introduced into the host specific phases, such that we can infect the bacteria with that phases. Now, these phases do not possess the intracellular biochemistry necessary to express this gene remain dark. So, the bacteria which are not going to have the capacity of expression of these genes will remain dark, and the one which is going to have the capacity of expression, they are going to be seen in this color. That means on transfer of this Lux gene to the host bacterium during the infection results in the light emission that can be seen or detected easily by luminometers. And the capacity of this bacterial luminescence can be detected, that means luminometer can detect of about 100 cells per hour. And the specificity of this assay depends on phase specificity. Example, bacteriophage P22 is specific for Salmonella typhimurium detection. Then coming to the electrical impedance biosensor. So here the electrical impedance biosensor is also going to be of electrochemical impedance spectroscopy. Okay, in general, impedance is the apparent resistance in an electrical circuit or to the flow of alternate current equivalent to the actual resistance to the direct current. When organisms, that is microorganisms, grow in a culture medium, so here you can see the culture medium, they metabolize substrates of low conductivity into products of higher conductivity, and hence the decrease in the impedance of the broth is going to occur. So what is meant by this impedance means the impedance is going to be of the response of an electrochemical system to an applied potential is called as impedance. That means the frequency is going to depend on this impedance can reveal underlying chemical process in the cells. Okay, so once again, the electrochemical impedance is the response of an electrochemical system, that is nothing but our cell, to an applied potential is going to be called as impedance. So now this whatever the finally resistant is going to decrease with growing cell numbers, then the impedance of the growth culture is going to be measured. The curvatures are produced for species and strains, and even the mixed cultures can be identified by use of specific growth inhibitors. Now, the time required for measurable changes to occur depends upon inoculum size that we are using. Then, all the detection limits here are theoretically a single cell if conditions are going to be acceptable. The main advantage of this electrical impedance biosensor is it is going to be widely used in food microbiology for pre-detection of shelf life of foods and a diversity of bacterial growth modeling studies for specific pathogens like E. coli, Campylobacter, etc. And moreover, this impedance can have the other applications also in finding the spoilage of microorganisms in defermentation process. Then impedance can be used to categorize the frozen orange juices, and it has been employed to detect the starter culture failure within 120 minutes, and it is going to be a quick method to detect the coliforms as well as the fecal coliforms in the meat. So that's how in food industry this method can also be used to predict the shelf life of the pasteurized milk also. So we are having the so many advantages of this electrical impedance biosensor. So with this, we have finished the two methods of detection of pathogens in the foods. That is, one is the conventional method, which we have discussed in the previous part. And in this part, we have discussed about the different types of rapid detection methods used to detect the pathogens in the food. Thank you.